Hello, everyone, and welcome to InBio's latest webinar on molecular allergy diagnostics, where today we'll be discussing InBio's innovative products and resources that can serve the diagnostic industry. I want to send a quick thank you to everyone for taking time out of their busy day to join us today. Certainly excited to present this information. We've got a great lineup of speakers today, starting with Dr. Sabina Wunschmann. Sabina is Vice President of Allergen Manufacturing and R&D with InBio. She's located in our U.S. headquarters, which is in Charlottesville, Virginia. Also joining us today is Dr. Nicole Brackett, Senior Scientist with InBio. Nicole is also located in Charlottesville, Virginia as well. We have a very special guest speaker today, Dr. Lorenz Aglas. Lorenz is Senior Scientist at the Paris-Lodron University of Salzburg. We're both excited and honored to have Lorenz joining us today, where he will be discussing his research on basophil mediator release testing using human IgE monoclonal antibodies. And I'm Beatty Sturgill, Senior Business Development Manager with InBio, and I'm with other team members here in Charlottesville. InBio has been serving the allergy diagnostics developers and manufacturers over the past 10 years, primarily by providing high quality, reliable, and readily available products and services. And the focus of today's webinar is going to be discussing InBio's molecular approach and how we can help improve new and existing diagnostic platforms. And one of the tools of that molecular approach is InBio's purified allergens. So Sabina will be presenting on that. Sabina will be going in depth on our purification process, which is done under an ISO 9001 certified quality management system. She'll also be sharing details of our extensive quality control and validation measures. But the goal of Sabina's talk will be to share an understanding of the manufacturing process, but also the benefits of using InBio's purified allergens in a diagnostic platform. Another tool in our molecular approach is a new product to InBio, which is Human IgE Monoclonal Antibodies. These are unique and exclusive to InBio, and they're derived from allergic patients that have a clinical history of allergic disease using proprietary methods. Nicole will be speaking to this and presenting on this. She's going to provide some background on the technology, as well as the QC measures we put in place. But the goal of Nicole's talk will be sharing an understanding of the significance of these human IgE monoclonal antibodies, as well as their roles in diagnostics and other applications. And then finally, Lorenz will discuss his critical research and updates on basophil mediator release testing using InBio's human IgE monoclonal antibodies. And we'll close the webinar with any time that we have remaining with an open discussion and a Q&A session. You can post comments or questions at any time during the webinar by clicking the Q&A button. And you can just post your questions there. You can post them anonymously, and we'll get through as many as we can by the end of the webinar. But with that, I will turn it over to Sabina to discuss purified allergens.
Thank you, Beatty. I will share my screen. All right. So over the next few minutes, I would like to talk about allergen manufacturing, validation and QC, and also introduce a few new allergen products. So the main advantage of molecular allergy diagnostics is that it allows to establish a patient-specific sensitization profile to individual allergens. And this is done using purified natural or recombinant allergens rather than allergen extracts. Having that unique patient-specific sensitization profile then allows the clinician to make informed decisions about risk assessment, cross-reactivity, and the best choice of treatment. For accuracy and precision of molecular allergy diagnostics, it is essential to use validated purified allergens of highest quality. Quick shout out on the Molecular Allergy User's Guide, which was just published last month, actually in March, by the European Academy of Allergy and Clinical Immunology. And here you can find comprehensive information on major allergens and their clinical relevance. This is a view of our main lab at our Charlottesville, Virginia headquarters, where all of our manufacturing takes place. The allergen manufacturing, QC, and product development all happens exclusively in-house using ISO 9001-2015 certified standardized procedures. Using these procedures, we manufacture over 100 purified allergen products from food, dust mite, cockroach, animal pollen, and venoms and mold. And our main product categories are natural allergens, recombinant allergens, and low-tox allergens, which can be either natural or recombinant allergens and have a very low endotoxin level, which makes it ideal for human and mouse cellular studies. A quick rundown on natural versus recombinant allergens. The advantages of natural allergens include that it is a natural form of the sensitizing allergen. It has the relevant mixture of isoforms and the natural occurring post-translation modifications. Potential downsides of natural allergens are that the abundance may be low in the source material. There may be some inherent variability of the natural source materials, and the purification may simply not be feasible. Also, trace amounts of other allergens from the same source may co-purify and need to be removed. An advantage of recombinant allergens is that as you're using a clone-specific allergen sequence, you will receive a standardized reagent of a defined quality that is also readily scaled up. Potential downsides of recombinant allergens include that the expression and purification may not be feasible, that there are trace amounts of host proteins that may co-purify and need to be removed. You have to decide on expressing one isoform of the allergen, and the folding and the post-translation modifications may differ from the natural allergen. Validation of the purity of the allergens is done by SDS page and mass spectrometry. We have an in-house Thermo Fisher Q-Exactive available. On the right-hand side, you can see the SDS page on the reducing conditions showing the 15 kilodalton recombinant ALT1 monomer. On the left-hand side is the mass spec analysis of recombinant ALT1 with the sequence coverage on top and the purity by relative abundance of over 99% below. We also validate the purified allergens for IgE binding and for IgG binding in allergen-specific ELISA assays. Here we are comparing two different versions of a recombinant ALT1, one expressed in Pichia and one expressed in E. coli. As you can see on the right-hand side that we got an excellent correlation for IgE binding between these two recombinant ALT1 products. And in the allergen-specific ALT1 ELISA assay, we also got excellent reactivity of both recombinant versions of ALT1 and with almost superimposed reactivity curves. Validation of the shelf life is done by a 5-year real-time stability study, where we are mostly looking at consistency of the protein concentration using OD280 and ELISA assays, but also for purity and stability of the allergen on SDS page. Again, this example of recombinant ALT1 shows that over a 5-year time frame, we got a consistent protein concentration with no drops and no signs of degradation by SDS page. Now I want to highlight a few new products that we have recently added or that we are working on. We've recently added few new tree nut allergens from almond, Brazil nut, pecan, pine nut, and pistachio, and are currently working on 3 native hazelnut allergens that will be released soon. The SCS page in the middle shows a side-by-side comparison of our tree nut 2S albumins. All of these tree nut 2S albumins are considered major allergens that are very stable and have a high resistance to digestion and food processing. And IgE binding to these tree nut 2S albumins is associated with a higher risk for severe allergic reactions, which makes the 2S albumins important biomarkers for the diagnosis of tree nut allergy. We also are working on new allergens for wheat and buckwheat. If I can draw your attention to the tree diagram in the middle. Based on solubility, the wheat proteins can be classified broadly into a salt or water soluble fraction, which are the non-gluten proteins, and the salt or water insoluble fraction, which are the gluten proteins. On the non-gluten protein side, we have several allergens, including the alpha amylase inhibitors, which were purified using a natural wheat extract and validated by mass spec as triad 28, 29, and 30. Also included in the non-gluten fraction is a wheat LTP triad 14, which we express as a recombinant allergen. On the gluten side of wheat proteins are the gliadins, and we purified alpha, beta, and gamma gliadin from the natural wheat extract, which was also validated by mass spec as triad 20 to 21. The omega-5 gliadin, or triad 19, was purified and expressed as recombinant allergen. We also added 2 new buckwheat allergens, FAGI2 and FAGI3, and those were both expressed as recombinant allergens. I'm excited to introduce a new product line of food protein standards. These are derived from food source flours using ISO 9001 standardized aseptic procedures. These are low-tox proteins with a defined allergen content, which is either validated by ELISA or mass spec. Applications for these products include in vitro research, such as mouse models of asthma, T-cell studies, food allergy research, but also can be used as reference standards for allergen standardization purposes. We have currently available the following food protein standards from peanut, hazelnut, pistachio, and pea flour, and are working on a soy flour, wheat flour, and whole egg protein standards. In my last slide, I have a comparison of 2 of these food protein standards. On the left-hand side is our low-tox peanut flour protein, which is validated by ELISA and contains defined amounts of RH1, 2, 3, 6, and 8, as well as a total protein concentration, which is validated by BCA. The hazelnut flour protein on the right-hand side is validated by mass spec and has defined allergen concentrations by relative abundance of CoA 1, 8, 9, 11, and 14. And this is also validated by BCA for total protein concentration. And with that, I want to thank you for your attention and thank the Allergen Group for their product development and their manufacturing of all these array of different purified allergen products. Back to you, Beatty.
Thanks, Sabina. Certainly a lot of good information on the purified allergens, and it's really exciting to see those food protein standards in the pipeline. But with that, I think we can turn it over to Nicole to provide some information on our Human IgE Monoclonal Antibodies.
Okay. All right. Thank you. So thanks for the introduction. So again, I'm Nicole, and I'm going to talk about our Human IgE Monoclonal Antibodies. So first, I'm just going to give a bit of background. Studies of human IgEs and the epitopes to which they bind on allergens overall are relatively limited. And in part, that's due to the polyclonal nature of human IgEs, as well as their low concentration in human sera. And then it's also due to the challenge of identifying and culturing IgE-producing B cells. However, with the recent development of hybridoma technology, it's allowed for the identification, amplification, as well as generation of IgE-producing cell lines. So this is shown in the figure on the right-hand side. So blood is taken from an allergic patient. The B cells from that blood are then expanded in a 96-well plate. The IgE-producing B cells are then identified and selected by ELISA, and those cells are fused with a myeloma partner. And then finally, the hybridoma cells are positively selected for those that are expressing IgE. So the panel of human IgE monoclonal antibodies that we have here at InBio were derived from 32 fusions or 16 allergic patients. And those patients have a history of allergic disease, which includes asthma, food allergy, atopic dermatitis, allergic rhinitis, etc. And primarily those allergic patients were also children between the ages of 3 and 18. So here's a chart of InBio's current panel of human IgE monoclonal antibodies. These human IgEs have specificity to 9 food allergens, 4 inhaled allergens, and alpha-gal. So on the left-hand side, the chart shows our wide variety of human IgEs that target peanut allergens. So these include RH1, RH2, RH3, and RH6. The chart in the center shows our human IgE monoclonal antibodies that target the different food allergens. And I specifically want to highlight the recent addition of 2 human IgEs, 16D9 and 10H8 at the top, which target the carbohydrate alpha-gal, which is associated with red meat allergen. And then finally, on the right-hand side, we have our panel of human IgE monoclonal antibodies that target allergens from inhaled allergens. So these are from dust mite, cat, as well as dog. In order to validate our panel of human IgE monoclonal antibodies, we run a variety of tests. So one, we measured the purity of the human IgEs, and this was done using SDS-Page. So this gel is just a representative example of our peanut allergen panel. So these samples were run under reducing conditions and stained with Kamasi blue. And then the gel shows the heavy and light chain bands. So again, this was done for our full panel of human IgE, which overall showed a purity of more than 90%. And for the majority, we saw a homogeneous heavy chain at about 75 kilodalton, and then a light chain at about 25 kilodalton. We also validated the human IgE using immunoblotting. So this was specifically to look at the specificity of our human IgE for their target allergen. So in this slide, I'm showing 3 representatives. But again, this was done for the full panel. So on the left-hand side, we investigated human IgE 3B10, which is targeted to peanut allergen RH1. In the center panel, we have human IgE 2F10 targeted to dust mite DERPy2. And then on the right-hand side, human IgE monoclonal antibody 9H7, which is targeted to JUGR1 from walnut. So for the 3 of these, the gel on the left-hand side shows the target allergen protein under both non-reducing and reducing conditions. So under reducing conditions with the disulfide bonds broken. And then the right-hand side, the gels that are outlined in red indicate the binding of the human IgE monoclonal antibody to those target allergens. Next, we looked at or investigated both the specificity and reactivity of the human IgE monoclonal antibodies using ELISA. So again, here are some representative plots, the nice sigmoidal ELISA plots for several of our human IgE monoclonal antibodies. These are dose response curves for the human IgEs targeted to either our purified or purified natural or recombinant target allergens. For this panel, all of the allergens were our natural allergens with the exception of JUGR1, which is shown in the bottom right-hand corner, which was our recombinant allergen. But the overall takeaway here is we had nice sigmoidal curves and showed good specificity and reactivity. Next, we measured the concentration of our human IgE monoclonal antibodies using ImmunoCAP. So we measured both the total and as well as the allergen-specific IgE levels for each of the IgE. In this case, I'm again showing just a representative chart of some of our monoclonal antibodies, our human IgEs, but we did this for the full panel. And if you're interested in seeing the full panel, I've included the link to our website, which is shown in the bottom right-hand corner. So what we see here, we measured our specific IgE for each of the human IgE monoclonal antibodies, total IgE, and if available, we also measured extract IgE. We ended up standardizing each of the human IgE monoclonal antibodies to 50,000 total IgE in order to compare the different monoclonal antibodies to one another. The main takeaway here is that the specific IgE was relatively high. So it ranged from about 468 to over 66,000 kilounits per liter, which is exceptional compared to the levels that we would see from human sera, which would have specific IgE levels normally in the range of about 1 to 10 kilounits per liter and at best 100 kilounits per liter. So these were pretty exceptional. The one that I will highlight with an asterisk is human IgE monoclonal antibody 14G12, which targets peanut allergen RH3. So that had a specific IgE level of 468, which was relatively low compared to both the total and extract IgEs, which are closer to 50,000. So in order to further investigate that difference, we then used the ImmunoCAP to look at the reactivity of several of the peanut human IgE monoclonal antibodies using both our natural and recombinant allergens. So in this case, we are looking at 4 of our human IgE monoclonal antibodies, so 14G12, 20M10, 38B7, and 13D9. And again, this was using the extract caps, so these are labeled peanuts. Those were from Thermo Fisher. Also using Thermo Fisher's recombinant RH3, RH6, RH2 caps. And then also using our natural or recombinant purified allergens, which were biotinylated and coupled to streptavidin caps. So what I'm highlighting here in red, we're showing that we had much higher specific IgE levels, specifically looking at 14G12 at the top when we used our natural RH3 as compared to what we saw for the recombinant RH3 cap from Thermo Fisher. So in this case, the natural RH3 resulted in a specific IgE of about 35,000, which was comparable to the extract value of about 31,000. So the takeaway in this case is that the recombinant peanut caps may ultimately lack certain IgE epitopes. So this stresses the value, again, of using our natural allergens in this case. Our human IgE monoclonal antibodies have a number of different diagnostic applications. So they could be used for quality control as well as validation of are purified natural or recombinant allergens for molecular testing and diagnostics. Also, the human IgE monoclonal antibodies could be used as potent alternatives to human sera for the validation of novel allergy diagnostic systems. They could also be used to identify clinically significant allergens as well as evaluate cross-reactivity. And finally, the human IgE monoclonal antibodies can be used for epitope mapping to look at cross-reactivity, which overall could have applications in both diagnostics and immunotherapy. To date, several pairs of human IgE monoclonal antibodies have been identified that bind non-overlapping epitopes on dust mite derpy2, FeLD1 from cat, and peanut RH2. And the figure at the bottom shows the first authentic structure of a human IgE epitope. So this shows human IgE monoclonal antibody 2F10 bound to derpy2 from dust mite. Here I want to highlight the different publications from which this data was extracted and also where further detail is explained. I also want to acknowledge the scientists here at InBio who were responsible for the expression, purification, and validation of the human IgE monoclonal antibodies. And then I also want to thank all of our collaborators. So those include collaborators from Vanderbilt University, Michigan State University, University of Salzburg, as well as Amsterdam University Medical Center. And thank you all for listening. And with that, I will turn it back over.
Thanks, Nicole. It's certainly interesting and great to see all the hard work that's gone into developing the human IgE monoclonal antibodies. And really interesting to see the data on the specific IgE levels. But with that, I think it's a great segue to turn it over to our guest speaker, Lorenz, here to discuss his research on basophil mediator release testing using the human IgE antibodies.
Thank you for the introduction, and also thank you for having me. Let me quickly share my screen. Okay, it should work now. So I have the opportunity now to talk about the applicability of the human IgE monoclonal antibodies that Nicole just mentioned, and especially in biological assays. And I'm talking now about a basophil mediator release assay. So just to show you quickly about our experimental model, how this works, we are using this basophil cell line, which has the high IgE binding Fc epsilon receptor 1 on the surface expressed. And we can sensitize the cells then with either the human IgE monoclonal antibodies or with IgE from patient serum. And then we stimulate the cells with the respective allergen or allergen source to induce cross-linking and then degranulation and mediator release. In our assay, we are measuring beta-hexosaminidase, an enzyme that can be easily measured in the supernatant as surrogate for histamine, for example, because histamine is less easy to identify or measure in the supernatants because of its short half-life. Here on the right-hand side, I'm showing the different kinds of human IgE monoclonal antibodies that we've obtained from InBio. We got 3 antibodies specific for derpy2, the major house dust mite allergen, 3 specific for FeLD1, 5 for the peanut allergen RH2, and then also a GALDI1 specific IgE antibody that we've used as a control. I will talk about this later. So the first thing we were interested in was to identify which pairs of monoclonal IgE antibodies are actually working in order to induce cross-linking and then degranulation of the basophils. And we had 3 different combinations of DRP2 specific IgE antibodies. And as you can see in this graph here, with the monoclonal antibody concentration on the x-axis and the mediator release on the y-axis, that only one combination actually resulted in a pronounced mediator release. I have to clarify one thing here. On the x-axis, we have the specific monoclonal antibody concentration and not the total concentration that Nicole mentioned before. We've repeated these assays in total 3 times and then also analyzed the area under the curve. And here you can see that this release is also significant. For the FeLD1 specific IgE antibodies, we could show that 2 combinations here in pink and in blue are actually resulting in a pronounced mediator release. And if you have a closer look on the green combination, this is actually even more sensitive compared to the pink combination, meaning already at lower monoclonal antibody concentrations, a mediator release is already induced. For the Rh6 specific antibodies, out of 10 possible combinations, 8 actually worked. 2 did not work. They were basically, the release was almost as baseline or even lower than the baseline. And we had 1 combination here shown in green, which induced the highest mediator release. As a control, we wanted to investigate if sensitization of the cells with just a single monoclonal antibody would also result in degranulation of the cells. As you can see here, only the combination, so for the case of the DRP2, only the combination induced mediator release, whereas there was a flat line for the individual sensitizations. In case of FeLD1, it was the exact same thing. However, for Rh2, we also observed we found one monoclonal IgE antibody that induced the degranulation by itself, so on its own. And of course, we were then interested in understanding how this could occur. And therefore, we were having a closer look at the structure, at the sequence of Rh2. And you can see that on this sequence of Rh2, we have this repetitive motif starting with aspartic acid and ending with serine. And this motif occurs in total 3 times throughout the structure. If we compare this to Rh6, both of them have a sequence similarity of about 60%. We can see that this repetitive motif is not being found on Rh6, meaning that this monoclonal IgE antibody specific for Rh2 might not recognize Rh6. I will come back to this later. As an additional control, we've paired the specific monoclonal IgE antibodies with a non-specific IgE antibodies or with a monoclonal IgE antibody with a different specificity. So in this case, the GALDI4 monoclonal IgE antibody. And here we've observed the same pattern as shown for the individual sensitization controls. That's why I will not go further into detail here. Because the next thing that was very interesting for us to investigate was the specificity of the monoclonal IgE antibodies. And here in the case of the DRP2 specific antibodies, we've used on the one hand recombinant and natural DRP2, then 2 different kinds of house dust mite extracts, and then natural and recombinant DRF2. So this is the homolog from DRP2 in Dermatophoidis farinae, and also the DRP2 homolog in Plomio tropicalis T2. And as you can see, the homologs are not really recognized, or stimulation with the homologs does not really induce mediator release, whereas for the DRP2 containing samples, they were relatively similar. I also have to point out here with antigen concentration, we actually are talking about the DERP2 concentration within the extracts, for example, or the respective DERF2 or PLOT2 concentration. So this was actually standardized to the DERP2 concentration. Regarding FeLD1, we've also used recombinant and natural FeLD1 and then a cat hair extract. And we've observed here with this combination that all three samples induced relatively similar mediator release when we stimulated the sensitized cells with this. Regarding RH2, we observed that we get the same signal for RH extract and RH2. However, RH6 did not really induce mediator release when we have our cells sensitized with this combination of specific RH2 antibodies. And this despite of this very high homology that I've pointed out before. If we compare this now to serum sensitization, meaning that we've sensitized the cells with a serum pool from our peanut allergic patients, we cannot really distinguish between the signal induced by RH6 from the signal induced by RH2. This is just to further point out that the specificity of the RH2 specific monoclonal IgE antibodies is very advantageous in this case. We also compared then the sensitization with the monoclonal IgE antibodies with the serum pool sensitization in terms of sensitivity. And here we could show that this combination in pink for the DERP2 specific monoclonal IgE antibodies is very similar to sensitization with a serum pool of house dust mite allergic patients. Then, however, for RH2, the situation was a little bit different. There, the serum pool was more sensitive than the combinations. And with this, I'm coming already to my conclusions. So we could confirm the biological activity of these human IgE monoclonal antibodies. The sensitivity is very comparable to serum pools. However, the antigen specificity is highly enhanced. So they are very specific. And on top of that, we've identified an anti-RH2 specific monoclonal IgE antibody that was able to induce cross-linking on its own. And in general, I want to point out that these human IgE monoclonal antibodies it's a lot easier to work with them. They're better to be standardized because of the limitations of the serum. For example, limited amounts of serum that are available, then the reproducibility of serum, and also the polyclonal character and the allergen specificity of the serum, which can hardly be controlled for. And if you're further interested in our research, we've recently published our results in Frontiers of Immunology. And with this, I would like to thank our collaborators from InBio and Scott Smith from Vanderbilt Medical University Center, and also Professor Ronald van Ree from Amsterdam UMC. Thank you for your attention.
Great. Thanks, Lorenz. That was a great presentation and a really great example of one of the applications of human IgE monoclonal antibodies and the impact that it can have on the in vitro diagnostic community as well. With that, we can wrap up here. I'd just like to share a summary slide that is one of InBio's missions to support in vitro molecular diagnostics. And as we discussed today, the tools that we can provide include the diverse panel of inhalable and food allergens. These are clinically relevant allergen proteins that, as Sabina demonstrated, have undergone extensive validation and QC testing. Excuse me. Another tool is the Human IgE Monoclonal Antibodies. Again, this is unique and exclusive to InBio. And as Nicole spoke to, they're derived from allergic patients with a clinical history of allergic disease. And it is really a breakthrough technology. that Lorenz pointed out, has diverse research and diagnostic applications, including the basophil release testing. But a lot of our products and services are built on scientific expertise. We have over 25 years in allergen purification and research. And all of that culminates into how we are able to help molecular allergy diagnostic developers and manufacturers with their platforms. With that, we can get to the discussion section and Q&A. Again, feel free to post any questions by clicking the Q&A button and typing them there. And we'll get through as many as we can in the next little bit. But we will be emailing out this webinar and contact information and follow-up information to everyone in the coming days so that you'll have a copy of this webinar. And as always, feel free to reach out. But with that, let's turn it over to... So Sabina, we can start with some purified allergen protein questions. The first one we have coming in is, which expression systems are used for recombinant allergen manufacturing?
Yeah, so it's predominantly E. coli and Pichia pastoris that we use for the recombinant allergens. But when we worked on some COVID expression, we also started doing mammalian expression using HEX cells. And we also have a CHO cell system that we can use.
Mm-hmm. Great. Another question we have is, do you conduct custom allergen development? I'm assuming that means for if we don't have an allergen protein in our product portfolio, do we do custom development?
Yeah, we've done that for several allergens and actually also non-allergens. Mm-hmm. So we would just research, come up with a best expression system that we can use, or if it is natural, the best way to purify it, and then do that as part of a contract research project. We decide on timeline and we usually with a recombinant, we would start with E. coli, or sometimes with both Pichia and E. coli, just to save time, see which expression system gives us the best results, and then move on with, decide on one to move on with for the validation of the protein.
Mm-hmm. Mm-hmm. Yeah, and just to add to that, something that we found that's important to molecular diagnostic companies is we have a policy in which we can send evaluation vials of an allergen to test in the platform, and then we can hold that same lot of, a certain milligram quantity. We can place that on hold while the evaluation testing is being done and completed. And we're happy to do that for customers. Sabina, one other question here. How are the allergens delivered? What state are they in?
Yeah, so they're all frozen liquids, and our standard vials size is 0.25 milligrams. The only product that we have that's actually lyophilized is our molecular reference standards that we have for 4 dust mite proteins. And that's has defined, it's validated by mass spec, and it has a smaller amount of 20 micrograms, I think, of lyophilized allergen that is really done for allergen standardization purposes as a reference standard for DERP1, DERF1, DERP2, and DERF2. Those are the 4 that we have. All the others are frozen liquids at this point.
Great. Nicole, we have several Human IgE questions here. Starting with, for the Human IgE levels measured using ImmunoCap, what accounts for the differences between total and specific IgE?
Yeah, so good question. Overall, the ratio of the total to the specific IgE tends to vary by allergen as well as by the individual human IgEs. And that difference between total and specific could reflect the differences in the IgE binding to the monoclonal anti-IgE that's used in the total IgE caps, versus the human IgE binding to the recombinant allergen that Thermo Fisher is using in their specific IgE caps. So I guess the overall value, though, or the takeaway is that I would still stress that even if the specific IgE levels were relatively low, the levels that we measured for our human IgE monoclonal antibodies were still much higher than what we would see from patient sera.
Mm-hmm. Mm-hmm. Great. I think this next question would be a great follow-up. In the peanut allergen analysis, what might account for the difference in specific IgE levels between your natural and recombinant allergens? They have given the specific example of monoclonal antibody 13D9 to RH2.
Also a good question. So I think that points back to what I had mentioned that we were seeing also with Thermo Fisher's recombinant allergens and their specific IgE caps. So this may maybe due to a lack of certain IgE epitopes included in those recombinant allergens, which again stresses, I would say, the value of using our natural allergens in order to measure specific IgE.
Mm-hmm. Mm-hmm. And do we have any information on the cross-reactivity or overlapping epitopes of the human IgE monoclonals?
We do. So I would say if we have epitope information or specificity information for any of our human IgEs, that's something that we'll include with the product information for those human IgEs on our website. So for example, with the epitope information, like I'd mentioned, we know that there are non-overlapping epitopes for that bind the human IgEs, specifically on allergens DERP2, FeLD1, and RH2. It was also recently identified the sequence and structure of the binding epitope of human IgE 2F10, which binds to DERP2. So that was recently published in 2022 in a paper in PNAS Nexus. So if we have that information available, it will be included in the product information for those. And then in terms of specificity or cross-reactivity, we also have that information, for example, for a number of our peanut allergens. And I guess I would stress that since the human IgEs are a novel product, we're always collecting new data. So as we get new information on specificity, cross-reactivity, or overlapping epitopes, that's something that we'll continue to update.
Great, thanks. So still with the human IgE monoclonal antibodies, the question here is, so they bind only to a specific epitope. Do you think that they can be an alternative to human sera to characterize extracts?
Interesting question. So I would say that is application dependent. So it depends on what you want to do with them. So again, the value is that the specific IgE values of our human IgEs are much higher than you would get with patient sera. So that would be valuable depending on what your application is. If you were going to look at something like specific epitopes, then it would be valuable to use our human IgEs versus patient sera. But yeah, overall, I would say it really depends on what your application is. So this is a potent alternative for a number of applications, but we also recognize there might be times where human sera might be the valuable or the value approach.
Yeah, Lorenz, I don't know if you have anything you want to add to that, but I know, in your slides and your presentations and in your conclusion, you mentioned that, the human IgE monoclonal antibodies do make a great alternative due to limited availability of sera, reproducibility of the antibodies, as well as the against the polyclonal nature of sera. So I don't know if you have anything you'd like to reiterate with that.
Yeah, exactly. I can totally agree with Nicole. It's definitely complementing sera at this point. If we will really be able to replace serum completely with the monoclonal IgE antibodies, I don't know yet. I think the future will show.
Great. Thank you. Another one for you here, Nicole. Why the large discrepancy of monoclonals to FeLV1?
Good question. So I think that goes back to the first question, just that we see, quite a range, and it really varies by allergen and again by individual human IgE.
Great. Well, thanks, Nicole. So Lorenz, several questions came in during your presentation there. Have you screened the biological activity of IgE monoclonal antibodies specific for any other allergen yet?
Yes, good question. So at the moment, we are also screening human IgE monoclonal antibodies specific for RH6. And why is this highly relevant? Because in peanut extracts, for example, we have at least 3 conglutens, which are all about the same size. So 15 to 17 kilodalton, we have there RH2, we have RH6 and RH7 as well. And of course, it would be highly relevant to differentiate the signals induced by each single one of them. If we're talking now about quality control or long-term storage conditions of extracts, if we really want to ensure that the biological activity is still given after a certain period of time, then we could do this with the specific monoclonal IgE antibodies. And this is the reason why we are screening now these specific antibodies against RH6. We also have obtained there some really nice results, and yeah, I can only say that the specificity of this monoclonal antibodies is really great.
Next question is, are you planning to analyze the biological activity with other assays as well?
Yes, so we definitely want to validate the monoclonal IgE antibodies also in a mast cell line, for example, in addition to the basophils. And what I'm highly interested in is using these monoclonal antibodies in sorts of inhibition assays, where you can really determine then, so usually in these inhibition assays, if it's now, I don't know, a FAB assay, facilitated antigen binding assay, or in an inhibition variant of the mediator release assay, you usually always rely on really high amounts of sera or serum pools to do this assays, to perform these assays. And of course, if you would have these monoclonal antibodies available, this would make things a lot easier.
Lorenz, we can start with you on this question, but Nicole, you may want to chime in. What can the human IgE mAbs be used for?
I think Nicole mentioned this already, so definitely for epitope mapping, this is highly good. And then, of course, inhibition approaches, as I've already mentioned. And yeah, and in general, investigating or characterizing AIT extracts or skin prick test extracts, I think this is highly relevant.
Yeah, I think that covers most of it. Yeah, the only other additions that I had in the list of applications were just, validation, quality control, purified allergens, as well as validation or testing for any novel diagnostic systems.
Lorenz, a chat question coming in. Can you get a prozone effect with the cellular activation with pairs? Seemed FeLD1 showed this.
Yes, so quickly to remind everyone what the prozone effect is. This is at really high concentrations that the signal drops again. And here we need to differentiate that in our assays with the basophil cell line, we have on the one hand titrated the concentration of monoclonal IgE antibodies, and on the other hand, we also titrated the antigen we've used to stimulate the sensitized cells in the end. And when we were screening just the sensitization with the monoclonal antibodies, we are really reaching a nice plateau, a top plateau, which remains rather stable. So even if you add more IgE antibodies to the cells, they will probably just end up in the supernatant and not become attached, and you will also not detach IgE then if you add simply more to this. But I think this prozone effect that was mentioned here in the question is referring to the antigen concentration, and this is something that we've observed very frequently also when using sera. And the reason for this is, so why is the signal, why is the mediator release dropping at very high concentrations of the antigen? And this is because of monovalent occupation of the IgE antibodies, meaning the antigen binds then just to one single antibody, and cross-linking is not occurring anymore because there's just so much of the antigen available. And this is then the reason why the signal drops again, and then you usually get these really nice bell-shaped curves.
Great, thank you. Let's see, the next question here is, maybe I missed this, but this is for you, Lorenz, but have you tested human IgE monoclonal antibody 38B7 against RAH2, inducing cross-linking and mediator release on its own as well on RAH6?
Yeah. So this is a really good question. And I have to say, the first time when we saw these results, of course, my initial reaction was this can't be. Just a single antibody cannot induce degranulation on its own. And of course, we then kept repeating the assay over and over again. And it was always the same pattern. We've used different aliquots, and we can definitely ensure that this is correct. And the question here, if we've also screened the 38B7 upon stimulation with RH6, yes, we did this. I think I've also shown this graph. And this was with the comparison of the different antigens. And there, the RH6 did not induce degranulation, which again points out in the direction that it's really this repetitive motif that is being recognized by this antibody, which is not present on RH6.
Yeah, interesting. Another question we have here is, do you have human monoclonal antibodies against Aspergillus or Alternaria? We do not currently, but you know, as Nicole mentioned, this is a novel product line that we're continuing to grow and work on. So it's possible that we may pursue adding these to the product line down the line. But with that, that's all the time we have questions for today. Want to really send a thank you for everyone attending today, taking time out. Special thanks to Lorenz for joining us. It was really great to have you on to discuss your research, and you provided a lot of valuable content. So thank you for joining. And thanks to Sabina and Nicole as well for presenting all that information. But again, we'll be sending out this recording and some follow-up information to everyone. And the InBio team will be at IACI here in, I believe, next weekend, June 9th through 11th. If anyone is in Hamburg and wants to stop by and see us, we'd love to meet with you. But with that, I think we'll close it out. Thanks, everyone.